Hello and welcome back to Science of Medicine. Across the next few lectures, I'd like to talk to you about cardiac physiology. And I'd like to do this from a standpoint of looking at it both structurally as well as electrically and pump-wise. And then we'll talk about some clinical applications of this. This is a very important topic, as you can imagine, for people that are studying medicine and physiology. So let's get right to it. So the heart is located generally on the left side in the thoracic cavity, nestled within the lungs, and it's basically two pumps that are side by side. There's one that's on the left and one that's on the right. And the reason for this is because when the left side of the heart, which is by far the stronger side, pumps blood out to the body, that pump is not strong enough to bring it back and also send it through the lungs. So I need a separate pump on the right side to take blood from a low pressure side and then give it a little bit more pressure to be able to pump it through and to do the gas exchange in the lungs. Now, both the left and the side are bipartite chambers. The antechamber into which blood is flowing are called the atria. If it's on the left, it's called the left atrium. If it's on the right, it's called the right atrium. And then the more muscular portions, which are called the ventricles, left on the left, right on the right. And each of these has valves associated with it. So on the right side, the valve between the right atrium and the right ventricle is the tricuspid valve. And then from the right ventricle out the pulmonary artery is the pulmonic valve. On the left side, between the left atrium and the left ventricle is the mitral valve, and between the left ventricle and out the aorta is called the aortic valve. So I have four valves, four chambers. Ideally, this blood is going to move in a forward direction, and I'm going to take the blood that comes into the heart and pump it out or provide it force to go out from the heart. It's going to move out in blood vessels away from the heart. Those are called arteries. The transport blood vessels back to the heart are called veins. There is a connection between those two, and those are capillaries. You may remember when we talked about the hematologic system that those capillaries that go throughout the body are very, very small, allowing basically about one erythrocyte at a time to move through. Now the purpose of this is to transport this blood, the nutrients, heat, cells like from our immune system, all throughout the body, collect the waste and send it to the appropriate disposal system. For example, like I mentioned before, the carbon dioxide that's going back to the lungs, but also toxins that are going to the liver or kidneys to be eliminated or detoxified. In order to do this, you need to actually have pressure. This is called the arterial blood pressure or just blood pressure. And that pushes this through because as these arteries move throughout the body, they split, split, split. And as a result of that, there's pressure drops along the way. If I look at these arteries and veins, they're quite different structurally. The arteries are much more massive in terms of their wall structure. They are comprised of not only an endothelial or inner layer, just like the veins, but also several other layers that are muscular and elastic. And those allow the arteries to expand or dilate as well as contract. And that elasticity in that holds energy, potential energy, if you will, that as the blood courses through that artery and expands the artery, on that recoil, it will provide some additional pressure. Now, these different layers on the arteries are so thick that the blood vessels that are called arteries, especially the larger ones, require their own blood supply. And that seems to be a little counterintuitive, but just like the heart muscle requires its own blood supply, so do some of these large arteries. And so they have the vasa vasorum, which actually supply nutrients to the blood vessel itself. Veins, on the other hand, tend to be more floppy. They do have a very thin layer of smooth muscle. They, of course, have that endothelial layer, but they really don't have the musculature or elasticity of an artery. And so they're holding much low pressure blood flow, and they require help to get blood back to the heart for pumping. Now, if it lies above the heart, that's no problem because gravity helps. But if it's below the heart, then you need to actually use the skeletal muscles in addition to the small amount of smooth muscle in those veins to help move that blood backwards to the heart. So another component of the heart is the heart muscle itself. Now, cardiac muscle is similar to skeletal muscle in a lot of ways. I still have myosin walking along actin filaments. I still have a sarcoplasmic reticulum, which is releasing calcium. I still have calcium binding with troponin C to be able to move the troponin I out of the way in order to do this. But I'm going to have a little different things with my cardiac myocytes. One of them is that the electrical signal will propagate much more quickly during depolarization. And that's facilitated by what are called intercalated discs. These discs lie at the ends between muscles that allow for rapid movement of ions. The other thing is that my contraction occurs via intrinsic, what are called pacemaker cells. These initiate depolarization without acetylcholine. So that means that I don't have to have a nerve that synapses with the cardiac muscle that releases acetylcholine, binding with an acetylcholine receptor and opening up a sodium channel. Instead, I have these pacemaker cells that will, with a certain time, depolarize automatically. I can modulate how fast this occurs via either hormones, something like adrenaline or norepinephrine, or also via the vagus nerve, which is a parasympathetic nerve that actually will slow the heart rate. Contraction also occurs with a little additional depolarization changes, and that has to do with two types of sodium channels, a fast and a slow, and a calcium sodium channel. The net effect of this is that the depolarization, which is normally fast, in cardiac myocytes is slower. And the purpose of this is to allow only forward transmission of the electrical signal and creation of a refractory period so the heart can't be re-excited. This keeps you out of certain electrical arrhythmias. Just to remind you, muscle, such as cardiac muscle, is a type of excitable cells. That means that if I have a concentration gradient of charged species, I will have a membrane potential difference. Cardiac myocytes are no different in this. This is accomplished by a sodium-potassium ATPase, which will pump sodiums out and potassiums in. There are also leakage channels that will actually can leak either sodium or potassium. Now, I could calculate what the potential difference across the membrane would be if I know the concentrations and permeabilities of various ions. This is given by the Goldman equation. But I want to talk about the general shape for an action potential or for excitable cells, and then modulate that and show you how that's a little bit different with cardiac myocytes. So first off, the depolarization, that rising phase, you'll have some depolarization which is allowing those ion channels, sodium channels, to open up. Now in this case, for just a regular action potential, I have a single sodium channel that opens up. Sodium channel opens up, sodiums rush in from the outside of the cell. That changes the cell's potential to be more positive on the inside, and that then is what leads to that increased depolarization, that increase in potential value. After a fairly brief period of time, about a millisecond or so, that sodium channel will close. I also will activate voltage-gated potassium channels, which will open up and cause potassium to flow out of the cell. That leads to a peak in the potential and a repolarization where the sodium is no longer flowing into the myocyte and the potassium is flowing out. That channel will also close, and then I can pump out and pump in with the sodium potassium ATPase to restore that resting membrane potential. Now I mentioned it's a little bit different for these pacemaker cells. I don't have this signal coming from a nerve. Instead, what I have are these cardiac myocytes that are firing at a certain rate, generally about 1 time per second. And that leakage is occurring from this such that the membrane potential is going to then rise spontaneously. Now, as I mentioned to you, I have sodium channels, I have fast sodium channels, slow sodium channels, and a sodium calcium channel that are also doing this. I will then still have the potassium channels, which will repolarize, and then I can go through this same system again. We will talk more in detail about the electrical signal and then also the pumping that results as a result of this in subsequent videos. I hope this has been a little helpful to you, and I'll see you soon. As always, you're welcome to subscribe or like or give me feedback about this, and I'll talk to you soon.